Imagine every single cell in your body as this tiny, highly specialized chemical factory, a factory managing like trillions of simultaneous reactions, perfectly efficient, all while maintaining order and powering everything you do.
It really is the ultimate system of logistics and efficiency. That constant critical management of inputs, outputs, energy,
Yeah.
it's what sustains life.
If those factories stop.
Well, if those chemical factories stop running smoothly or if their energy balance gets thrown off, everything halts, sometimes, instantly. It really is the definition of cellular control.
Okay, let's unpack this incredible system then piece by piece. This deep dive is focusing entirely on metabolism, that fundamental process dictating how cells acquire, manage, and, well, use every single unit of energy.
Exactly. We're diving into sources that define these complex metabolic pathways, the universal thermodynamic rules that govern energy, the critical role of enzymes, the biological machinery, and the highly sophisticated regulatory systems that keep the whole network in balance.
So our mission is really to take these core concepts.
And show you the elegance of how they interconnect to define life itself. And, crucially, how even small failures can lead directly to disease.
Right. So let's start with the baseline definition. When a biologist says metabolism, what exactly are they talking about?
Well, at its simplest, it's just the sum of all chemical reactions occurring within a cell. It's not one single pathway. It's the entire interwoven chemical landscape, really.
Okay, the whole landscape.
Yeah.
And it's organized.
Definitely organized
Yeah.
into two major functional categories, which are like eternally linked in this inescapable partnership: catabolic and anabolic.
Catabolic and anabolic. Okay.
Catabolic pathways are, you could say, the demolition crews of the cell. They break down large, complex molecules, things like proteins or carbohydrates, into smaller units.
And that releases energy.
Crucially, yes. This process releases chemical energy, which means these reactions are labeled as exergonic. They give off energy.
Got it. Exergonic energy exiting. And anabolic.
Anabolic pathways are the construction workers. They take those small, simple building blocks, the ones from catabolism maybe, and build complex macromolecules, new proteins, new DNA, new cell walls, whatever the cell needs.
But construction takes energy, right?
It requires effort, yeah.
Yeah.
So these pathways require energy input. We label them as endergonic reactions. They need energy put in.
Okay, so exergonic releases, endergonic requires,
Okay.
and that sets up.
The central tenet of the cell's entire economy: energy coupling.
Energy coupling, explain that.
That's really the core idea. The energy released by the catabolic demolition crew, that exergonic energy, is immediately channeled and utilized to power the anabolic construction project, the endergonic ones.
So they have to happen together.
Pretty much. Without this constant coupling, the cell would just collapse. It wouldn't be able to build or maintain itself. It's continuous dynamic partnership, really.
Okay, so before we even get into the molecules and the pathways and the enzymes, we have to talk about the rules, right? The universal constraints.
Absolutely. Metabolism doesn't matter if it's bacteria or humans. It has to obey the laws of physics. And the big ones here are the laws of thermodynamics.
Okay, let's start with the first one.
The first one is probably the easiest for us to grasp. The first law of thermodynamics. It's the principle of energy conservation.
Energy can't be created or destroyed.
Exactly. Only transferred or transformed. The chemical energy locked in the food you eat doesn't just vanish. It changes form, maybe into heat or kinetic energy for movement, or it gets stored as chemical energy in ATP. But it's all accounted for.
Okay, that seems straightforward enough. What about the second law? That one sounds more complicated.
Yeah, the second law of thermodynamics is where life really has to work hard. This law governs entropy.
Entropy, disorder, randomness.
That's it, the measure of disorder or randomness in the universe. The second law basically dictates that the total entropy of the universe always, spontaneously, increases. Systems naturally trend toward maximum disorder. Chaos, essentially.
But wait, if you look at a cell,
Yeah.
it's incredibly ordered,
Yeah.
right? Highly structured, complex. That seems like the opposite of disorder.
It is. It's the very definition of localized low entropy.
So how does that work? Isn't that violating the second law? How can a cell maintain order, let alone grow, if disorder has to increase?
That's the brilliant distinction. It's about the system versus the surroundings. Cells maintain that extraordinary level of biological order only through constant massive energy input.
They have to fight it.
They have to continuously fight that universal trend toward internal entropy. They do it by importing and processing energy, and crucially, they do so by increasing the entropy of their surroundings. When they harvest energy, say from glucose, they release heat, which is disordered energy, and simple disorganized waste products like carbon dioxide and water. So the order inside the cell is maintained, even increased, but only at the cost of increasing the total disorder of the universe, the cell plus its environment.
Okay, so it's like an energy tax. They pay for their local order by creating more disorder elsewhere.
So that's a great way to put it. An energy tax the cell must constantly pay.
Right. This brings us to, well, the operational math of the cell. The way it calculates if a reaction is worth doing. That's free energy, or Gishi.
Exactly. Gishi stands for the change in Gibbs free energy.
Yeah.
And it is probably the most critical calculation for a chemist, or indeed for a cell. It represents the portion of a system's energy that is actually available to do work.
And the sign matters, positive or negative.
The sign tells the cell instantly whether a reaction is spontaneous and sort of profitable energetically or non-spontaneous and costly.
Okay, so if the change in free energy, the Ishi, is negative, delta G $7.
That means the products store less energy than the reactants did. Energy is released during the reaction. It's favorable, it's spontaneous, you call it exergonic. That's the demolition crew's payoff we mentioned.
Makes sense. And if the AKEs is positive, delta G all 1.
Then the reaction is non-spontaneous. It requires an external energy input to happen. It's endergonic. These are the reactions, like building complex structures, that absolutely need that energy coupling to a highly favorable energy source.
And that energy source, the universal currency the cell uses to, pay its energy bills, is ATP, adenosine triphosphate.
ATP's gang, yeah. It's the most readily accessible and versatile energy molecule around. It's kind of an intermediate, perfect for carrying energy from those catabolic reactions over to the anabolic ones.
And the energy release isn't like burning the whole molecule, right? It's specific.
It's very specific. The magic happens when the cell performs hydrolysis, uses water to break a bond on the terminal, the outermost phosphate group.
And do we know how much energy that releases? Our sources gave a number?
They did. That hydrolysis reaction is highly exergonic. It releases a standard free energy change of approximately negative 30.6 kilojoules per mole. That's a significant chunk of localized energy available immediately.
Wow. Okay, so how does the cell use that energy burst? It doesn't just explode, right?
No, exactly. It's much more elegant.
Yeah.
This is the mechanism of the cash transaction, if you like. When ATP is hydrolyzed, the energy isn't just released vaguely into the surrounding water. Often that released phosphate group doesn't just float away.
What happens to it?
Instead, it's frequently transferred directly onto one of the reactant molecules that needs activating for an endergonic reaction. This temporary attachment of the phosphate group creates what we call a phosphorylated intermediate.
Ah, so the phosphate physically attaches to the molecule that needs energy.
Yes, and that phosphorylated intermediate is now more unstable, more reactive, has higher free energy than the original molecule. This makes the subsequent originally endergonic reaction much more favorable. It basically forces the uphill reaction to proceed precisely when and where the cell needs it to.
So it's targeted energy transfer through a chemical change. That's clever.
It is. It forces chemical work. But what's really striking when you look at the full scope of cellular function is just the sheer variety of work that ATP drives. It's not just powering chemical construction.
Right. The sources mentioned 3 main types of work driven by ATP. We covered chemical work powering anabolism. What are the other 2?
Okay, first there's mechanical work. This is maybe the most visible use of ATP if you think big picture. Think muscle contraction.
Like lifting something.
Exactly. That large-scale movement is powered by the cyclical interactions of actin and myosin filaments. ATP binding and hydrolysis drives the shape changes, the conformational changes of those motor proteins that make the filaments slide.
And smaller scale.
On a smaller scale, it powers things like motor proteins walking alongside of skeletal tracts inside the cell, maybe dragging vesicles or organelles around. It's physical movement.
Okay, mechanical work. And the second non-chemical type.
Transport work. This is absolutely fundamental for survival. The classic crucial example here is the Texnia plus Texpain, the sodium-potassium pump.
That's in our cell membranes, right? Pumping ions.
Embedded right in the plasma membrane, yes. And it actively transports ions against their natural flow. It pumps 3 sodium ions out of the cell and 2 potassium ions into the cell, both moving against their concentration gradients, uphill.
Which requires energy.
For its constant energy input from ATP hydrolysis. Maintaining that steep electrical and chemical gradient across the cell membrane is absolutely essential. Without it, you can't generate nerve signals, you can't control cell volume properly. Basically, cells fail.
So that pump is a perfect example of ATP coupling forcing transport work.
Absolutely. Forcing those ions uphill against the electrochemical current.
And of course, ATP isn't like an infinite supply. The cell has to remake it constantly.
Oh, constantly. ATP is regenerated from ADP, adenosine diphosphate, and inorganic phosphate, pi, by capturing energy released during our catabolic processes, primarily cellular respiration.
Which includes glycolysis, the TCA cycle.
And oxidative phosphorylation, yeah.
Right.
We're talking about literally millions, maybe billions of ATP molecules being consumed and regenerated every single second in a highly active cell. It's an incredibly rapid turnover.
Okay, so if thermodynamics sets the rules what's possible, what releases energy, then enzymes are what the technicians, the workers who actually make the reactions happen fast enough.
That's a great analogy because even if a reaction is thermodynamically favorable, delta G dollars, it might still happen incredibly slowly on its own. Could take hundreds, even thousands of years spontaneously. Enzymes drop that timescale to milliseconds, sometimes microseconds.
Wow. So their primary role is speed.
Speed, exactly. Enzymes are biological catalysts. They dramatically accelerate reaction rates by lowering the activation energy.
Activation energy, that's the energy needed to get the reaction started, like pushing a rock up a small hill before it can roll down the big hill.
Precisely. It's the energy required to contort the reactant molecules into an unstable, high-energy transition state before they can become products. Enzymes provide an alternative pathway with a much lower activation energy hill.
And crucially, they aren't used up in the process, right?
Correct. They facilitate the reaction, but emerge unchanged at the end, ready to catalyze another one. And importantly, they don't change the overall energy difference, delta G1, between reactants and products. They just make it happen much, much faster.
Okay, let's look inside the enzyme itself. The key part is the active site, yeah.
The active site is the business end. It's a specific region, usually a groove or pocket on the enzyme's surface, where the substrate molecule the enzyme acts upon binds.
And the binding isn't just like a key fitting into a rigid lock anymore.
No, that model's a bit outdated. We now use the induced fit model. It's more dynamic. When the substrate binds to the active site, the enzyme itself subtly changes its three-dimensional shape.
It adjusts to the substrate.
Exactly. It tightens around the substrate, creating the highly specific enzyme-substrate complex. It's more like a glove adjusting to a hand than a rigid key in a lock.
Why does that shape change, that induced fit, matter so much?
Well, that optimal fit does a couple of things. It doesn't just hold the substrate. It can physically stress specific bonds within the substrate, making them easier to break. Or, if there are multiple substrates, it orients them perfectly relative to each other for the reaction to occur.
So the enzyme actively helps the reaction along by stressing or positioning the substrate.
Precisely. That mechanical stress or optimal orientation is what dramatically lowers the required activation energy. The enzyme is truly facilitating the chemical transformation.
But enzymes need to be controlled too. You mentioned the active site for the substrate. But there's another important site, the allosteric site.
Right. The allosteric site is often the control panel. It's a separate binding site, physically distinct from the active site. Regulatory molecules bind here.
To do what?
To either turn the enzyme on, turn it off, or maybe just fine-tune its speed. It's a major way the cell regulates enzyme activity without directly interfering with the substrate binding site.
Okay, and sometimes enzymes need a little extra help, right? Support staff.
Yeah, sometimes the amino acids making up the enzyme aren't enough to do the required chemistry. They need non-protein components to assist in catalysis. These are broadly called cofactors and coenzymes.
Okay, what's the difference between those two? They sound pretty similar.
They do sound similar, and they both help the enzyme, but they differ chemically. Cofactors are generally the inorganic components, typically metal ions.
Like iron or magnesium.
Exactly. Things like zinc, Textiv2 plus Wasava, iron ions. These charged particles can do chemistry that amino acid side chains can't easily manage, like helping to stabilize charges during a reaction.
Okay, so cofactors are inorganic ions and coenzymes.
Coenzymes are the organic helper molecules. They're often larger and more complex. They frequently act as temporary carriers, shuttling chemical groups, atoms, or electrons from one reaction or one enzyme to another.
Like delivery trucks.
Sort of, yeah. They cycle back and forth, picking up a payload from one enzyme and dropping it off at another. We'll talk more about key examples like text NAD plus oxis and text AD, which carry electrons, or coenzyme A, which carries acetyl groups.
And many vitamins are related to these.
Absolutely. Many B vitamins, for instance, are precursors that our bodies convert into essential coenzymes. That's why vitamin deficiencies can cause such widespread metabolic problems the coenzymes are missing, and enzymes can't function properly.
Right. Now, since enzymes are so powerful at speeding things up, the cell must have ways to slow them down or stop them when needed. Inhibition.
Definitely. You don't want these reactions running full tilt all the time. There are two main types of enzyme inhibitors we usually discuss based on how they interact with the enzyme.
Okay, what's the first type?
Competitive inhibitors. These molecules often look structurally very similar to the enzyme's normal substrate. They're mimics. They're mimics, yeah. And because they look similar, they can bind to the enzyme's active site. They compete directly with the real substrate for access. If the inhibitor gets in there, it blocks the substrate, and the reaction stops.
So it's direct competition for the main binding spot.
Exactly.
Yeah.
The outcome often depends on the relative concentrations of the substrate and the inhibitor. If you flood the system with substrate, you might overcome the competitive inhibitor.
Okay, and the other type?
The other main type is non-competitive inhibitors. These guys don't compete for the active site at all. They bind somewhere else on the enzyme.
Often at that allosteric site we mentioned.
Very often, yes. They bind to the allosteric site or another remote site. And this binding causes a change in the enzyme's overall three-dimensional shape. Its conformation changes.
And that affects the active site even though the inhibitor isn't bound there.
Precisely. The shape change is transmitted through the protein structure, and it physically distorts the active site. So even if the substrate can still bind, the active site isn't shaped correctly anymore to actually perform the catalysis. The induced fit mechanism is ruined.
So it inhibits function indirectly by changing the enzyme's shape.
Right. And unlike competitive inhibition, adding more substrate usually doesn't overcome non-competitive inhibition because the inhibitor isn't blocking the substrate binding site directly.
This idea of controlling an enzyme by binding somewhere else seems really important. That's allostery, right? And there's also feedback inhibition.
Yes, allosteric control, whether it's activation or inhibition by molecules binding to that separate site, is a key mechanism for rapid reversible regulation. But perhaps the most elegant control loop for an entire metabolic pathway is feedback inhibition.
Feedback inhibition. That sounds like the system regulating itself.
It's exactly that, self-regulation perfected. Imagine a multi-step assembly line making a product. In feedback inhibition, when the final end product of that pathway starts to accumulate, meaning the cell has made enough of it that end product molecule itself travels back.
Back up the assembly line.
Back up a pathway, yes. And it acts as an allosteric inhibitor for one of the enzymes much earlier in the process. Often, it inhibits the very first enzyme that's unique to that specific pathway, the committed step.
So the product shuts down its own production line when there's enough.
Exactly. If you've got enough inventory, stop the assembly line right at the beginning. It prevents the cell from wasting energy and resources, making something it doesn't currently need. It's incredibly efficient internal management.
All right, so we've got the thermodynamic rules, we've got the enzyme machinery. Now we need to connect them into the actual factory layout. Metabolism isn't just random reactions, it's organized into metabolic pathways.
That's right. It's a highly integrated highway system, not just a collection of isolated back roads. The basic organizational principle is usually a multi-step process, where the product of one enzyme-catalyzed reaction becomes the substrate for the next enzyme in the sequence.
Like a baton handoff in a relay race.
Exactly like that. One enzyme does its job, modifies the molecule, and passes it directly to the next enzyme in line. This ensures the whole process is directional and allows for multiple points of regulation along the way.
And these pathways, they have different shapes or structures, don't they? The sources mentioned a few common patterns.
They do. We see a few recurring structures. The simplest is probably the linear pathway. Glycolysis is the classic example here.
Glucose is going to pyruvate.
Right. You start with one molecule, glucose, a 6-carbon sugar, and through a series of sequential steps, it gets converted into 2 molecules of pyruvate, 3 carbons each. It's pretty much a straight shot from A to B to C and so on.
Okay. Linear. What else?
Then you have cyclic pathways. The most famous example, of course, is the TCA cycle, also known as the Krebs cycle or the citric acid cycle.
Why cyclic? What makes it a cycle?
In a cyclic pathway, one of the starting molecules needed for the first step is actually regenerated at the very end of the cycle. So the cycle can turn over and over again, processing new inputs continuously, as long as those inputs are supplied. It's like a continuously running engine that just needs fuel fed in.
Okay, linear, cyclic. Any others?
And then there are branched pathways. These are especially common in biosynthesis pathways, like when the cell is making amino acids or nucleotides.
Branched how?
An intermediate molecule partway through a linear sequence might serve as a starting point, a branch point, leading off to synthesize 2 or more different final products. The pathway splits.
So the cell can choose which branch to follow based on its needs.
Exactly. This allows for flexibility. Maybe the cell needs more of product X than product Y today. The regulation at these branch points is often very sophisticated, directing the flow of intermediates down the needed path.
Right. Now, all this chemical movement, breaking bonds, making bonds, transferring energy, it requires specialized carrier molecules, doesn't it? Like the delivery trucks of the cell.
Absolutely critical.
Yeah.
We need to talk about the key energy transfer molecules because they really define the direction of energy flow in metabolism. First up, text NAD plus text NADH.
Okay, what's its job?
Text NAD plus steed nicotinamide adenine dinucleotide is the primary electron carrier involved almost exclusively in catabolism, the breakdown pathways. The oxidized form, text NADD plus D, accepts electrons, and usually a proton, so it accepts a hydride ion, text NEF, released during the breakdown of fuels like glucose or fatty acids.
It becomes NADH.
Becomes the reduced high-energy form. Text NADH is essentially holding on to that captured chemical energy, primarily in the form of high-energy electrons, which it will later donate to the electron transport chain to make ATP. Think of it as capturing energy from fuel breakdown.
Okay, text NAD plus text NADH for catabolism. What's next?
Next is text FADH, Texter's FADH22. Flavin adenine dinucleotide. It's similar to text NAD+ in that it's also an electron carrier used in catabolism.
So why two different ones?
Well, text FAD tends to accept electrons from molecules that have slightly lower energy levels than those that reduce text NDT+. You see text FAD being reduced to text AVDH22 in specific reactions, particularly in the Krebs cycle and also during fatty acid breakdown, beta oxidation. It captures energy from slightly different steps than text NAD+ DOM.
Okay, so both text NADH and text FADH2 are carrying energy captured during breakdown destined for ATP production.
Correct. They're like charged batteries ready to power the ATP synthase machinery.
Now, there was a third one mentioned that sounded similar but had a different role. Text NADP plus dollar.
Ah yes, text NADP plus text NADP2. Nicotinamide adenine dinucleotide phosphate. This is really interesting and super important. Notice the extra P for phosphate. This molecule, specifically its reduced form text NADPH2, provides reducing power primarily for anabolism, for building things up.
Wait, reducing power. What's that exactly, and why is it different from the energy NADH carries?
Great question. Reducing power refers to the ability to donate high-energy electrons needed for synthesis reactions. Building complex molecules like fatty acids or steroids involves forming new carbon-carbon bonds, which often requires adding electrons, reduction. Text NADPH is the cell's main source of these electrons for biosynthesis.
So text NADPH waves carries electrons to make ATP, while text NADPH will largely separate even though they look similar. The cell keeps the pools of text NADH or collagen ADPH largely separate even though they look similar. Text NADH job is to be oxidized, or lose electrons, in the mitochondria to drive ATP synthesis. It's about energy generation. TET NADPH job is primarily in the cytosol, where it's oxidized, loses electrons, to provide the reducing power needed for anabolic reactions like fatty acid synthesis. It's about energy input for construction.
Why keep them separate? Why not just use NADH for everything? It's about control.
Yeah.
By keeping the primary catabolic electron carrier separate from the primary anabolic electron carrier, the cell can regulate breakdown and synthesis independently. It ensures that the drive to make ATP doesn't deplete the reducing power needed for building, and vice versa. It's like having separate bank accounts for operating expenses and capital investment.
That's a fundamental separation then. Really clever.
It's absolutely fundamental to maintaining metabolic balance.
Okay, any other key carriers?
One more major player is coenzyme A, CoA. It's different because it doesn't carry electrons. It carries acyl groups, basically, activated carboxylic acid groups.
The most famous being acetyl groups.
Exactly. When coenzyme A carries an acyl group, a two-carbon unit, it forms the incredibly important molecule acetyl-CoA. Acetyl-CoA is like a major crossroads in metabolism. It's the common molecule generated from the breakdown of carbohydrates via pyruvate, fats, and even some amino acids. And it's the main fuel input for the TCA cycle.
So CoA acts like a handle for carrying these important two-carbon units around.
Precisely. It activates the acetyl group, making it ready for reactions like entering the Krebs cycle or being used for fatty acid synthesis.
Okay, let's quickly summarize the central tension again, the flow between breaking down and building up using these carriers.
Sure. Catabolic pathways. Their job is to break down complex molecules. They are net releasers of energy. They generate the cash, take ATP, and the high-energy electron carriers destined for ATP production, text NADH22, think glycolysis, beta oxidation, fat breakdown, Proteolysis, protein breakdown. The goal is energy harvest.
And anabolic.
Anabolic pathways. Their job is to build complex macromolecules. They're net energy consumers. They use up large amounts of text ATP for energy, and they require that dedicated reducing power from text ADPH for the actual construction work. Think gluconeogenesis, making new glucose, fatty acid synthesis, protein synthesis. They consume the products of catabolism to ensure growth, repair, and maintenance, constantly fighting entropy, as we said.
Right, so the existence of all these pathways, catabolic and anabolic, using these carriers, that's amazing in itself. But maybe the real achievement is how the cell controls it all, right? How it prevents them from just running wild or, worse, running in opposite directions at the same time.
Absolutely. The control is incredibly sophisticated and multi-layered. We have to appreciate the emergent properties of the network. It's not just a collection of independent pathways. It's a deeply interconnected web.
Meaning a change in one place can affect things far away.
Instantly. Feedback ripples across the system. The sources give a good example. Imagine oxygen supply becomes low. Anaerobic glycolysis cranks up to try and make ATP.
But that produces lactate, right? Lactic acid.
Exactly. And if that lactate builds up, it lowers the cellular text pH, makes the cell more acidic. That change in text might be subtle, but it can dramatically affect the activity of dozens of other enzymes throughout the cell. Enzymes involved in completely different pathways because most enzymes have a very narrow, optimal text range.
So one pathway going into overdrive because of low oxygen can inadvertently mess up lots of other unrelated processes just by changing the pH.
Precisely. It shows how sensitive and interconnected everything is. A small perturbation can have widespread consequences.
So to manage this complexity and keep things running efficiently, cells use multiple levels of control. The sources mentioned 4 main levels.
Yes, at least 4 hierarchical levels of metabolic regulation, controlling both the speed and the direction of metabolic traffic, often simultaneously. Let's go through them, maybe starting with the fastest response.
Okay, level 1.
Level 1 is allosteric control. This is pretty much instantaneous regulation. We already talked about allosteric inhibition with feedback loops, but allosteric activation is just as important.
Binding at that separate site turns the enzyme on or off quickly.
Right. It ensures the metabolic factory floor responds immediately to changes in supply and demand. The perfect example is the regulation of a key enzyme in glycolysis called phosphofructokinase 1, or PFK1. It's a major control point.
PFK1 commits glucose to being broken down for energy. How does it know when to work faster or slower?
It acts like an immediate energy sensor for the cell. If the cell's energy levels are low, this is signaled by rising levels of text AMP, adenosine monophosphate, which forms when ATP is heavily used.
AMP means we need energy.
Basically, yes.
Yeah.
And AMP acts as a powerful allosteric activator of PFK1. It binds to the allosteric site, turns the enzyme on full blast, speeding up glycolysis to generate more text ADP.
And if energy is high?
Conversely, if text ATP levels are high, the cell has plenty of energy currency, then TEC-DP itself acts as an allosteric inhibitor of PFK1. It binds to a different allosteric site on the enzyme and slows it down.
So ATP inhibits its own production pathway when levels are sufficient. That's like the cell checking its bank account balance before spending more fuel.
Exactly that. It's
Yeah.
very elegant immediate feedback.
Okay, that's level 1, allosteric control. Level 2.
Level 2 is covalent modification. This is still quite fast, though maybe not instantaneous like allosteric control. It involves chemically modifying the enzyme itself. The most common way is by adding or removing a phosphate group. This is called phosphorylation, adding a phosphate, and dephosphorylation, removing it.
And specific enzymes do this adding and removing.
Yes, enzymes called kinases add phosphate groups, often using ATP as the phosphate donor, and enzymes called phosphatases remove them. Adding or removing that bulky, charged phosphate group physically changes the enzyme shape and consequently its activity. It acts like an on-off switch or sometimes a dimmer switch.
And this is used for signaling.
Very commonly used in signaling cascades.
Yeah.
The signal arrives at the cell surface, triggers a kinase, which phosphorylates an enzyme, changing its activity, which might then affect another enzyme. It allows for rapid, reversible responses to signals.
Okay. Level 3 involves communication across the whole body, right? Hormonal control.
Right. This is systemic regulation. Coordinating metabolism between different tissues and organs, especially important for managing fuel levels in the bloodstream, like blood glucose.
And the key players here are hormones like insulin and glucagon.
Those are the classic examples, acting antagonistically. Insulin is released when blood glucose is high, like after a meal. It signals a state of energy abundance. It tells cells, especially muscle and fat cells, to take up glucose from the blood, use it, and store the excess as glycogen or fat. It promotes anabolic processes overall.
And glucagon.
Glucagon is released when blood glucose is low, maybe between meals or during fasting. It signals an energy deficit. It primarily acts on the liver, telling it to break down its stored glycogen and also to synthesize new glucose, gluconeogenesis, and release it into the blood to bring levels back up. It promotes catabolic processes in the liver,
So they work in opposition to keep blood sugar stable.
constantly balancing each other out to maintain that crucial energy homeostasis for the whole organism.
Okay, that's 3 levels. What's the 4th?
The fourth level is a more structural form of control, compartmentalization. It's about spatial organization within the cell.
Putting different pathways in different places.
Exactly. By isolating specific sets of reactions and enzymes within different membrane-bound organelles, the cell can run potentially conflicting pathways simultaneously without them interfering with each other. It also allows for concentrating substrates or enzymes where they're needed.
It's like having different rooms in the factory for different processes.
Perfect analogy. It prevents the construction crew from getting in the way of the demolition crew or the raw materials for one process contaminating another. It ensures efficiency and provides another layer of control.
So let's map some key pathways to their compartments. Where does glycolysis happen?
The initial breakdown of glucose, glycolysis, occurs out in the main volume of the cell, the cytoplasm, or cytosol fluid part. It's relatively accessible.
But the main energy generation happens elsewhere.
Yes, the powerhouses, the TCA cycle and oxidative phosphorylation, where most ATP is made using oxygen, are located exclusively inside the mitochondria. Pyruvate from glycolysis has to be actively transported into the mitochondria to be further processed.
Okay, what about fats? Building them versus breaking them down.
This is a really critical example of compartmentalization for control. Fatty acid synthesis, building fats, happens primarily in the cytosol. And remember, it requires that specific reducing power molecule, TXNADPH, which is also mainly generated in the cytosol.
Okay, synthesis in the cytosol, breakdown.
Beta oxidation, breaking down fatty acids for energy, occurs almost exclusively inside the mitochondria. And this process generates TXNADH and TXNADH2, which can then feed directly into the mitochondrial electron transport chain for ATP production.
So building fats outside, burning fats inside. Why is that separation so important?
It's absolutely vital for regulation. It prevents a futile cycle where the cell is simultaneously making fatty acids in the cytosol and breaking them down in the mitochondria. The physical separation allows the cell, usually under hormonal control like insulin-promoting synthesis, glucagon-promoting breakdown, to tightly regulate whether it's in an overall energy-storing mode or an energy-releasing mode regarding fats. It keeps the opposing processes from canceling each other out.
That spatial organization really is another layer of elegant control.
It really is. It underpins the cell's ability to maintain that incredible order and efficiency we keep coming back to, minimizing wasted effort, maximizing useful output.
Okay, so understanding just how perfectly tuned these metabolic control systems are really shines a light on what happens when they go wrong, doesn't it? The medical relevance seems huge.
It's massive. Imbalances or defects in this vast metabolic network are directly responsible for some of the most common and devastating human diseases, things like obesity, type 2 diabetes. These are fundamentally metabolic disorders.
And sometimes the problem is genetic, right, from birth.
Yes. We see conditions known as inborn errors of metabolism. These are typically caused by mutations in a gene that codes for a specific enzyme in a metabolic pathway. If that enzyme is missing or defective, the pathway gets blocked.
And the consequence.
Often the substrate of the blocked enzyme builds up to toxic levels, or a necessary product isn't made. This can cause severe problems, especially if it happens early in development.
The sources mention specific examples like phenylketonuria, PKU.
PKU is a classic example. A defect in the enzyme that processes the amino acid phenylalanine. If it's not caught early and managed with a very strict low phenylalanine diet, the buildup causes severe intellectual disability. Maple syrup urine disease, MSUD, is another, affecting the breakdown of branched-chain amino acids. Again, toxic buildup leads to neurological damage without strict dietary control.
So these highlight how critical even one enzyme step can be in the whole network.
Absolutely.
Yeah.
Then you have the major systemic disorders that affect millions. Diabetes mellitus is arguably the biggest. It's fundamentally a disease of metabolic dysregulation, centered around glucose metabolism, but affecting fats and proteins too.
Driven by problems with insulin.
Either the body doesn't produce enough insulin, type 1 diabetes, an autoimmune disease, or the body's cells stop responding properly to the insulin that is there, type 2 diabetes, often linked to lifestyle factors like obesity. In both cases, that crucial hormonal control system we discussed is broken, leading to chaos in fuel management.
And problems can arise right in the powerhouses, too.
Definitely. Mitochondrial disorders are a group of conditions caused by defects in the mitochondria themselves, often due to mutations in either mitochondrial DNA or nuclear DNA coding for mitochondrial components.
It's affecting ATP production.
Directly affecting ATP production.
Yeah.
Since mitochondria are the main ATP factories, defects lead to severe energy shortages, particularly impacting organs with high energy demands: the brain, muscles, heart, eyes. The symptoms can be incredibly varied and debilitating.
Now, one area that's getting a lot of attention is how metabolism relates to cancer.
Yes, cancer metabolism is a huge and rapidly evolving field. It goes back almost a century to the observations of Otto Warburg.
The Warburg effect.
The Warburg effect, exactly. Warburg noticed that cancer cells have a very peculiar metabolic preference. Even when there's plenty of oxygen available, they tend to rely heavily on glycolysis for energy production, rather than sending pyruvate into the mitochondria for oxidative phosphorylation, which yields much more ATP.
That seems backward. Why would a rapidly growing cancer cell, which needs tons of energy, favor the less efficient pathway, glycolysis?
It does seem counterintuitive from a pure ATP yield perspective. Oxidative phosphorylation gives you maybe 30-something ATP per glucose, while glycolysis alone only gives you a net of 2 ATP.
So why do they do it?
The current thinking is that it's a strategic shift. While glycolysis is less efficient at making ATP, it can run much faster than the full mitochondrial pathway, and crucially, the intermediate molecules produced during glycolysis.
The molecules partway through the pathway.
Yes, those intermediates can be easily diverted, shunted off from energy production, and used instead as building blocks. Cancer cells need vast amounts of lipids, nucleotides for DNA, and amino acids for proteins to fuel their rapid cell division and growth.
So they're using glycolysis not just for ATP, but as a source of raw materials for construction.
Exactly. They prioritize rapid biomass accumulation over maximizing ATP yield from each glucose molecule. They're essentially running glycolysis in overdrive to supply the precursors needed to build new cancer cells quickly. It's a metabolic reprogramming that supports proliferation.
That's a fascinating and kind of sinister adaptation.
It is, and it's become a major target for potential cancer therapies trying to disrupt this altered metabolism, specifically in tumor cells.
Now, it's really important to remember this incredibly detailed map of metabolism wasn't just discovered overnight. It was built piece by piece over decades, centuries even, by many researchers.
Absolutely essential to acknowledge the history. The fundamental thermodynamic rules that govern everything we've discussed. We owe a huge debt to scientists like Josiah Willard Gibbs, who developed the concept of free energy, delta G, and gave us the mathematical tools to predict reaction spontaneity.
And the specific pathways. The Krebs cycle is obviously named after someone.
Indeed. Sir Hans Krebs famously elucidated the steps of the TCA cycle, the central hub of energy metabolism, in the 1930s, truly groundbreaking work that earned him a Nobel Prize. We'd also mention Albert Szent-Györgyi, who made crucial earlier discoveries about related organic acids and reactions that paved the way for Krebs's full cycle. He also got a Nobel for his work, partly related to vitamin C as well.
And Warburg, who we just mentioned for the cancer connection.
Yes. Otto Warburg's insights into cellular respiration and the altered metabolism of tumors were also Nobel Prize-winning work and remain profoundly influential today. Without these pioneers, and many others, meticulously working out the chemistry, the enzymes, the regulation, we simply wouldn't have the understanding we do today of this intricate cellular economy. Hashtag outro cell.
Wow. Okay, so if there's one big takeaway from this really deep dive, for me at least, it's the incredible elegance of it all, especially that energy coupling principle.
Yeah.
How the controlled destruction of one thing directly powers the construction of something else. And it's all orchestrated by these incredibly specific enzymes governed by layers upon layers of tight regulation.
It really is. Understanding metabolism is, in a very real sense, understanding the fundamental processes of life itself. It's the source of our energy, our growth, our maintenance, and, as we saw, also the origin of many diseases when it goes wrong. The whole system is just built on this foundation of efficient management, from the tiny ATP molecule acting as currency all the way up to organelles acting as specialized factory rooms.
So let's leave our listeners with something to think about. A final provocative thought, maybe building on those control mechanisms. We talked about feedback inhibition, how the end product of a pathway can shut down an early enzyme. Very precise internal regulation. Now, we also talked about chronic diseases like diabetes, often linked to broader hormonal issues, which current therapies sometimes address with, well, fairly blunt systemic effects.
Right. Hormones affect many tissues.
So, considering how powerful and localized feedback inhibition is within the cell's own machinery, could future pharmacological research leverage that principle more directly? Could we design drugs that don't just mimic hormones, but instead act like highly specific allosteric regulators? Maybe drugs that subtly adjust the sensitivity of a key enzyme to its own feedback inhibitor within a specific target tissue.
Ah, so using the cell's own internal inventory control system to fine-tune metabolism locally.
Exactly. Could that be a way to, say, gently tweak the balance between catabolism and anabolism in muscle or liver cells without causing the major systemic side effects you might get from broad hormonal intervention? Using the cell's intrinsic logic against itself, almost.
That's a fascinating direction. Really bridging systems biology, understanding the network with targeted drug design and personalized medicine. How to make those subtle, precise adjustments.
Right.
Definitely something worth exploring further.